Hello students, in this module we are going to be learning the process called as blastulation. Now when we talk about blastulation students, this is the structure which will be formed called as blastula. Let's see how it is formed. Now here in this structure students, you can see after the cleavage division, at the end of cleavage division, a 16 cell stage is formed called morula. Now in blastulation, morula starts absorbing uterine milk and forms an outer layer of cells which is called as trophoblast. The arrow pointing at the trophoblast. Cells start rearranging in the morula. The cells we call as blastomeres. So blastomeres start rearranging. Why? Because there's a fluid getting inside the embryo. So the cells, they kind of push towards an end. Cells rearrange, thereby resulting in the formation of a cavity which is called as blastocele, and we call this embryo which is having a cavity called blastocele. This embryo students is called as blastula. And now let's see how exactly it is formed. Process of blastulation students is defined as rearrangement of the cells or blastomeres, resulting in the formation of what? A fluid filled cavity which is called as blastocele. So process of rearrangement of blastomeres resulting in the formation of a fluid filled cavity is called as blastulation. And what is the final structure which is formed? What do you call this structure? Blastula, or there are two other names to it students. It's also called as blastocyst or blastodermic vesicle. Both the names indicating a fluid filled cavity being formed. Now blastocyst students is this structure. And what is this structure? Outer layer of cells is called as trophoblast or trophoectoderm. Inside it, the blastomeres were pushed to a corner. That group of cell students is called as inner cell mass. All of these cell students which you are seeing in here, this is called as inner cell mass or embryoblast or embryonal knob, because it looks like a knob. A cavity is formed. This cavity students is called as blastocele or the segmentation cavity, and it's filled with what? It's filled with uterine fluid or uterine milk. The end where embryoblast is present, that group of cell is present students, that end is known as embryonic pole or animal pole, and the opposite end here is called as abembryonic pole. This is the structure students which we will be studying in details. Blastula called as blastocyst also. Now let's see, when we talk about the fate of the structures of blastocyst students, what exactly is the role of trophoblast or inner cell mass or the cavity. When we talk about inner cell mass students, this group of cells which you're seeing and we are calling as inner cell mass, this is the future embryo. That means the entire future embryo is formed from which structure of blastocyst? Inner cell mass, also called as embryoblast. Talking about trophoblast students, trophoblast has got important roles like this is the structure students which forms extra embryonic membranes. Now these extra embryonic membrane students, in human there are 4 of them, namely amnion, chorion, allantois and yolk sac. Now these extra embryonic membrane students are not going to be directly involved in the formation of embryo, but they are going to be helping embryo, supporting embryo in some way or the other. So all of these 4 extra embryonic membranes are formed from trophoblast. Secondly, trophoblast provides nourishment to the embryo. Thirdly, students, trophoblast is the structure which will help in the implantation in the uterine wall. Now students, this is the structure called blastocyst. In here, the cells which are shaded there, the cells you're seeing here, that means I'm talking about the cells which are towards the embryonic pole side or touching the embryoblast students. These cells are called as cells of Robber, which are the cells which are in contact with the embryonal knob or embryoblast, cells of Robber. Now, another important thing in blastulation, students, is the membrane around the blastocyst is now breaking up or degenerating. Which is this membrane or layer we are talking about? The transparent layer, which is the zona pellucida. And why is zona pellucida disappearing? It has to disappear now because now implantation has to take place. Blastula now increases in volume and size, and blastocyst stage is reached 5 days after fertilization. Now herein we talked about blastulation, students. We are going to be continuing with the process of implantation. Till then, take care.